क्लास ले रहा हूँ एनपीटीईएल वाली की, बाद में बात करते हैं। Okay, then we are going to start. I don't think anyone else is going to join just at the moment. I will just share my screen and let us get started. Okay, good evening everyone. Welcome to the week 8 PMRF-NPTEL session. Today we are going to go through the lectures and videos on neural system and special senses. During this particular week we have gone a bit back and covered what was left of the stretch reflex arc as well as neuromuscular junctions, and learned a bit more about the inner workings of the ear and how the hearing sense actually works. We are going to go through all of those today. So I will just briefly introduce myself. My name is Animal Chatterjee. I am a Prime Minister Research Fellow and PhD scholar at IIT Kanpur, and I am hosting these sessions on Tuesdays. So as you would know, these are problem solving sessions being held in collaboration between NPTEL and PMRF, in which we go through a previous year's run of the assignment questions of the NPTEL course that we are going through this time, and then try and solve the different assignment questions in order to understand how exactly we can tackle these problems in an exam situation, and as well as have a better understanding overall of the subject. So all of these sessions are recorded, and if you have not been able to join into the live session that we are going through now, then you can just go onto YouTube and this particular playlist where all of these sessions are being uploaded, and if you want to go through the session at a later point of time, you can do so at your own convenience. So to start off, I would like to ask if anyone here has any questions regarding the lecture material of this week. Then we can start off deciding and discussing on that, and then move on to the assignment questions that we are going to do. So anything that anyone wants to ask about this week's lectures and the videos? Okay, I guess there are no outstanding questions. So we will just go into the first question for this week. So how we do this here is that I will show you a question which is from my assignment from previous year, and I would like you to go through the question and try and answer which of the following options of the MCQ are correct in the chat. So you will put into whatever you think is the correct answer into the chat, and then we will discuss about the question and answer it. So the first question for today is: physiologically, testing stretch reflex is an important test when we go to a doctor because. Okay, Aruna is asking something. We can actually go through that first. I think Vishwajit also has a question. Aruna is asking, sir, in auditory concerns, stereocilia or kynostelia is longer. So, as we'll go through the last part of today's PowerPoint, you will see that stereocilia are often drawn longer, but it does not matter if they are longer or not. They are the ones that are facing the. So, in the cochlea, if the cochlea is this tube-like structure, it is normally a snail-like structure, rounded up, but if you stretch it out, it will form a tube-like structure. The stereocilia are the ones that are facing towards the channel. So any vibration is first felt by the stereocilia, and stereocilia are connected to the kynocelia via various small filaments. So once the stereocilia moves in a particular direction, it will actually pull or push the kinesterecile in the same direction. So we often say that stereocilia is the one which determines if and when the particular entirety of the hair cells will move in which direction, and depending on their movement, the particular cell is either turned on or turned off. That is, it will either have action potential or not have action potential. Is that fine, everyone? So it does not matter if the stereocilia is drawn longer. Often the case is that the stereocilia is longer, but it is not a hard and fast rule. It is just the one that is in front of the channel facing in the direction of movement and determines where the other cilia are going to move, in which direction. Is that fine? Yeah. I think Vishwajit also had a question.
Sir, about hair cell, I did not understand that concept. Could you explain that after the, after the
The hair
class?
cells part, which part did you not understand? Because we also have a video today that actually goes through how exactly hair cells function as well. So maybe we'll tackle it when it comes along, and then after that, if you have the question still, then we can discuss it. Is that fine?
Uh, okay, sir.
Yeah. Okay. So coming back to the first question we have, physiologically testing stretch reflex is an important test when we go to a doctor because the options are A, it is used to measure blood pressure, it is B, it helps to understand body temperature, and without using a thermometer it correlates the pathology of central and peripheral nervous system. It helps to measure your walking speed. So we have answers coming in. Aruna, Vishwajit and Jagruti, all of them say it is C, it correlates to the pathology of central and peripheral nervous system. Anyone else that wants to try and answer this? Okay. So the muscle stretch reflex is most commonly determined by in when you go to a clinic or a doctor via a hit on the patella. Patella is the bone that covers our knee joint, and you probably had already have had it done or have seen it somewhere that often doctors carry this kind of a mallet which has a triangular shape, and they hit it on the patellar bone, and if they hit it just right, you will see a involuntary knee jerk or a reflex action. This particular reflex action is often used to determine if you have any peripheral nervous disorders or not, and it is a very common test. The reason being that the entirety of the patellar knee jerk reaction is goes through the spinal cord. So here we have a bit of details about it. So we have a particular nerve, which is the 1A afferent nerve or neuron, which actually detects this particular stimulus. This stimulus is then projected directly to the dorsal. We get disconnected. I might have gotten disconnected. Hmm. Okay, so the internet is back. You can hear you can hear me again, right? I am audible again, right? Audible and the PowerPoint is visible, correct? Am I audible and the PowerPoint is visible? Can you just
Yes, sir.
okay? So we are going to talk about the knee jerk response or the patellar bone uh, reflex. In this particular test, what the doctor does is that he has a particular mallet that is shaped like a triangle and hits the patellar bone right here. If he hits it correctly, then a particular neuron, which is the 1A afferent neuron, is excited. This excited neuron then carries the reflex all the way back to our spinal cord. See here that this is a reflex action, that is the higher parts of the brain, the cerebrum or the cerebellum, are not informed about the entire motion till after it has occurred. So the entire processing happens within the spinal cord via one or two neurons. So the first neuron that is affected is a interneuron, which processes the information and conveys the information back to a extensor motor neuron, or the neuron can directly affect a flexor neuron. So this information is gone back directly to our muscles, and this essentially causes our knee to bend forward. So essentially we lift our knee in response to a hit on the patella. This is a particularly important test and very easy test to do to check our peripheral nerve responses. So if any damage has been done to the spinal cord, then this entire circuit will be hindered and you will not see a proper reflex. And that is why this is often done regularly as well as in case of accident patients to determine if there has been any injury to the peripheral nervous system. So physiologically, testing stretch reflex is important because it correlates the pathology of central or peripheral nervous system to the knee jerk reaction. So, any questions from this? Okay, moving on. Blank is a reflex arc that provides direct communication between the sensory and motor neurons innervating the muscle. So, which reflex arc is the one where the sensory and the motor neurons are directly connected? The options are monosynaptic reflex, bisynaptic reflex, polysynaptic reflex, or reflux. So you have four options A, B, C, D. You can put whatever you think is the correct one into the chat. So in case of which reflex do you think that there is direct communication between sensory and motor neurons? Monosynaptic, bisynaptic, polysynaptic, and reflux. So Vishwajit says that it is B, bisynaptic. Anyone else wants to try and answer this? Jagbut says it is A, monosynaptic reflex. Sibani also says it is A, monosynaptic reflex. Anyone else? Aruna says it is B, bisynaptic reflex. Okay, so let us look at it. So here we have a video on how exactly muscle stretch reflex works, and I will be playing it for you so we have a better understanding of the entire thing. Okay. Let me just search for it.
In this video, I'm going to talk about the muscle.
Is the audio clear? You can hear the video, right?
In this video, I'm going to talk about the muscle stretch reflex. The nervous system performs.
The audio is audible, right? Hello?
No, sir.
What about this time?
Many reflexes. And a reflex is a
And
response
now
to
it
a
is
stimulus that
audible.
doesn't require the involvement of.
Okay.
In this video, I'm going to talk about the muscle stretch reflex. The nervous system performs many reflexes. And a reflex is a response to a stimulus that doesn't require the involvement of consciousness. All reflexes have two parts. The first part is called the afferent part. Afferent. And that involves bringing information about a stimulus into the central nervous system. So there'll be some sort of receptor somewhere in the body that can detect the stimulus, and then there'll be some sort of neuron that brings that information into the central nervous system. The other part of a reflex is called the efferent part of the reflex. Efferent, which carries information away from the central nervous system to cause a response somewhere in the periphery. So there will be some sort of neuron that'll carry that information away from the central nervous system out into the periphery to cause some sort of response. Now, some reflexes, like the muscle stretch reflex that I'm just about to describe, happen on the same side, so that the afferent part of the reflex brings information in from one side of the body, and the efferent part of the reflex brings information back to that same side of the body to cause the response. Other reflexes, particularly those up in the brainstem, have an afferent limb that comes in on one side, and then efferent responses that come out to both sides. So there's some variety for how the information travels in reflexes, depending on the reflex. One of the simplest reflexes that's a good example, and that happens to be one of the most medically useful, is called the muscle stretch reflex. Muscle stretch reflex. If a skeletal muscle, like in this drawing, here's a skeletal muscle in the arm, if this is rapidly stretched, the muscle stretch reflex will cause it to contract very quickly after it's stretched, presumably as a protective response to prevent injury to a muscle from being stretched too rapidly. But let's go over the one that happens around the knee, that's also called the knee jerk, because most of us are probably familiar with this one. Because a lot of us, when we're in our doctor's office, have had the experience where we're sitting in a chair. So here I've drawn a person, and we're looking at their right side, and here's their trunk and their leg. And if you're in the clinic, often your doctor will take a little rubber hammer, and they'll take that little rubber hammer and they'll hit you right below the kneecap. And to your surprise, when they hit you below the kneecap with a little rubber hammer, your leg will often kick out without you telling your leg to kick out. There's this involuntary response of the leg kicking out to the stimulus of the rubber hammer hitting you just below the kneecap. So why does this happen? Well, the place that your doctor is hitting you with a little rubber hammer is not actually in the kneecap itself, but it's in the tendon that's just below the kneecap. So let me draw that here in orange. And that tendon hooks onto the bones in the lower leg. And connected to the kneecap on the other side from the tendon is a large group of muscles in the front of the thigh. And when your doctor hits you in that tendon, it actually stretches this large group of muscles. Because for just a moment, the little rubber hammer bends this tendon, and that pulls on the kneecap like this, and that pulls on this muscle, and it stretches it. Now, it doesn't stretch it very far, but it does stretch it rapidly. And there are receptors in skeletal muscle that can detect muscle stretch. I'll just write a big R here to represent one of the receptors. And there are lots of these receptors spread out throughout all of the skeletal muscle in the body. And these receptors are called muscle spindles. Muscle spindles. And here's a drawing of a muscle spindle. So here's a skeletal muscle, and they've magnified this little receptor. And we won't go into the details, but there are these specialized little fibers inside the muscle spindle that get stretched when the rest of the muscle gets stretched. And then there are neuron axons that are wrapped around these special fibers that can detect that stretch of these fibers and send that information back into the central nervous system. So that these axons that are leaving the muscle spindle here will travel back through nerves of the peripheral nervous system, and then they'll enter either the spinal cord or the brainstem. And these are somatosensory neurons that tend to have their somas and ganglia close to the spinal cord or the brainstem. And since these are neurons carrying information into the central nervous system, we can call them afferent neurons, and they make up the afferent part of the muscle stretch reflex. Let me just write that out. That for the muscle stretch reflex, the afferent part are the somatosensory neurons. Somatosensory neurons. Inside the central nervous system, like here in the spinal cord, these somatosensory neurons carrying that muscle stretch information, that information about the stimulus, are going to form an excitatory synapse. So let me just draw a little plus sign here to represent an excitatory synapse with another neuron whose soma is in the central nervous system. And this neuron is going to send an axon out through nerves of the peripheral nervous system back to the same muscle that was stretched. And it's going to synapse on and excite skeletal muscle cells in that same muscle, causing the muscle to contract, causing the response. And the neurons that synapse on and control skeletal muscle cells are lower motor neurons. Lower motor neurons. And for the muscle stretch reflex, the lower motor neurons make up the efferent part of the reflex that causes the response of contraction of the muscle that was stretched. In the video where we went over the motor unit, we talked about lower motor neuron signs that can appear with abnormalities of the lower motor neurons, and we talked about hyporeflexia, meaning a decrease in the muscle stretch reflexes. And I think you could see why that would happen. If the lower motor neuron is not able to communicate with the muscle, then it can't tell it to contract in response to the stimulus of muscle stretch. But it turns out that you can also have diminished muscle stretch reflexes if there's a problem with these somatosensory neurons bringing information about the muscle stretch back to the lower motor neurons. So that if there's a problem with either the lower motor neurons or the somatosensory neurons, you can have a diminished muscle stretch reflex. And it turns out this is true for all reflexes. If there's a problem with either the afferent part of the reflex, bringing stimulus information into the central nervous system, or if there's a problem with the efferent part of the reflex, carrying response information out to the periphery, a problem with either the afferent or efferent part of a reflex can cause a diminished or lost reflex, because both parts have to be working for the reflex to occur. Now, one important thing to notice about reflexes is how all of this just occurs down here, in this case in the spinal cord, or it could occur in the brainstem if it was a brainstem reflex. But the higher parts of the nervous system, the cerebrum, where a lot of the higher functions of the nervous system, like cognition, emotion, and consciousness, they don't have to get involved for a reflex like this to occur. And this is the reason we say reflexes are responses to stimuli that don't require the involvement of consciousness, because the wiring tends to occur at these lower parts of the central nervous system and peripheral nervous system without having to involve the higher parts of the nervous system way up in the cerebrum. Now, this is all you need for the muscle stretch reflex, but there is another part to it that isn't necessary but does add something, because it turns out that while this muscle is contracting in response to the stretch of the muscle on the opposite side, in this example, the muscle on the back of the thigh that bends the knee when it contracts, this muscle actually relaxes. So while the muscle on the front of the thigh is contracting, the muscle on the back is relaxing. The way this occurs is that this same somatosensory neuron that's exciting the lower motor neuron back to the muscle that was stretched is also sending axon terminals to other neurons, and it's going to excite those neurons, so I'll draw a little plus sign. But these neurons are actually inhibitory neurons, so they're going to form a synapse that's inhibitory. So I'll draw a little minus sign to represent that they're inhibitory. And what they inhibit are lower motor neurons to the muscles on the opposite side. So these lower motor neurons would normally be exciting the muscles here in the back of the thigh that would cause the knee to bend. But when they're being inhibited by this other neuron, these lower motor neurons aren't exciting that muscle in the back of the thigh, so it relaxes. Now this isn't necessary for the reflex to occur. You just need the afferent and the efferent part of the reflex for it to occur. But because this muscle, when it's contracting, isn't fighting against this muscle on the back of the thigh since it's relaxing, that does increase the response. So there's more straightening of the leg at the knee and kicking outward. And a lot of the reflexes in the nervous system have some similarities to this sort of setup, where you can almost think of a balance between responses that the nervous system can choose from, and that the reflex tips the balance in favor of a response in one direction. In this video, I want to introduce
Okay, so I hope that clears what exactly you're talking about when we are discussing the muscle stretch response. We'll go through a bit here as well. So when we are talking about reflex action, we essentially mean that it is a particular response to a stimulus which does not involve the higher cognitive functions of the cerebrum or cerebellum, and thus can be tremendously fast and is often used in cases where we are in situations of getting bodily harm. You would obviously have seen situations in which you are touching a hot pan or have touched something that hurts you, and instantly you take your hand back. That is the situation where the reflex action takes place, where there is not enough time for the entire signal to be processed by the higher consciousness, and this is because the entirety of the response goes through via the spinal cord. So I will just sum up in brief what exactly reflex action and the stretch response or the stretch reflex arc is. First, we have a stimulus. This could be you touching a hot pan, holding your hand over a flame, which is then detected by a receptor. This receptor then passes the information on to a sensory neuron. The sensory neuron goes directly back to the spinal cord, where it can either synapse directly onto a motor neuron, or it can synapse onto a interneuron, which is essentially a buffer neuron, which then synapses back to a motor neuron. So sensory neuron could go directly to a motor neuron, as you can see right here. The motor neuron carries the signal back to the skeletal muscle, which asks us to actually pull our hand back from the flame or the hot material. This essentially is via the somatic motor neuron, which is also known as the efferent neuron. An efferent neuron synapses onto the target cell, which in our case is the skeletal muscle, but many cases include in which the target cell can be, or target tissue can be, a gland which is asked to release hormones, and then we have the response. So if this is the general situation, then if the sensory neuron goes to the spinal cord and synapses directly onto a efferent neuron or a motor neuron, then this is called a monosynaptic reflex, because there is only one synapse between the sensory neuron and the efferent neuron. So in our case, this is a monosynaptic reflex. So if we come back to the question, is a reflex arc that dash is a reflex arc that provides direct communication between sensory and motor neurons innervating the muscle? The answer is a monosynaptic reflex, because there is if there is direct communication, that is the sensory neuron is directly synapsing onto the motor neuron, there is only one synapse involved in the entire process, and thus it is known as a monosynaptic reflex. Is that fine? Is that okay?
Yes, sir.
Okay. The next question is just the opposite. The reflex arc involving the presence of one or more interneurons is called a monosynaptic reflex, a bisynaptic reflex, a polysynaptic reflex, or a reflux. So there are four options, A, B, C, D. You can choose whatever you think is the correct option. Aruna says it is C, a polysynaptic reflex. Anyone else? Vishwajit also says it is C, polysynaptic reflex. Anyone else wants to try and answer this? Jagruti and Sibani both say it is C, a polysynaptic reflex. Okay, so you are all correct. So in the case when the, so here's another example, you have touched a cactus, say. So in that case also, because of the reflex action, you pull your hand back. In that situation, it could be that the sensory neuron takes up the information that there is something harmful to our body close by and relays it via a relay neuron or an interneuron. So it synapses onto an interneuron which processes the information and then sends the information back to the motor neuron, and then the motor neuron can actually cause the muscle to contract and retract your hand back, and this information is processed by the brain only after the reflex action has already taken place, because this is much faster than relying on information being traveled on information being sent to the brain and coming back and informing the motor neuron to do something. So a reflex action is one where the higher motor centers are not involved and the spinal cord or the brain stem can take the decision to retract or contract a muscle or release hormones as and when required. Is that okay? So you have an interneuron, then it is a, so coming back to the question, if there is an interneuron involved, there will obviously be more than one synapse, and thus this particular kind of a reflex arc is known as a polysynaptic reflex, in which you have multiple reflexes. In this particular case, multiple synapses. In this particular case, you have a synapse here and a synapse here. So in this case, you have two synapses, and it is known as a polysynaptic reflex. Is that okay?
Yes, sir.
Okay. So next, the stretch reflex is also called, options are a bistatic reflex, a myotactic reflex, a reflex, or tendonitis. Vishwajit says it is B, myotactic reflex. Aruna also says it is B, a myotactic reflex. Jagruti also says it is a myotactic reflex. Anyone else? Okay. So yeah, you are correct. The muscle stretch reflex is known as a myotactic reflex. If you just divide the word up, myo means muscle, tactic means movement. So if you essentially just convert muscle stretch, it will be converted into myotactic. It is just a name that we use to determine the muscle stretch reflex. Okay. The next question is blank innervate extrafusal muscles fibers and blank innervate intrafusal fibers. The options are alpha motor neurons, alpha and gamma, gamma and gamma, gamma and alpha. So Aruna says it is B. Alpha motor neurons innervate extrafusal fibers and gamma motor neurons innervate intrafusal fibers. Vishwajit also says it is B. Alpha innervates extrafusal, gamma innervates intrafusal. Jagruti also says it is B. Anyone else? Okay. So yeah. So what is the difference between an extrafusal fiber and an intrafusal fiber? So essentially, all of our muscles that take part in contracting and help in movement, all the skeletal muscles, essentially are extrafusal muscles. These are these broad muscles that form the tube-like structures and are made out of muscle fibers. These contract to actually help move our various limbs and body parts. But there is a small percentage of the muscle that is known as the intrafusal muscles. These are small muscle fibers which have a different physiology as compared to extrafusal muscles. These are broader in the middle as shown right here and are present in between spaces in the entire muscle. And these are essentially sensory elements that help detect the stretch or the contraction or relaxation condition of the muscle. So for example, if you, this has been discussed in the lecture as well, if there is any condition in which your muscle is stretched, say your hand is being hit by something like this, even before the force is moved, the muscle comes back to its original position, as if someone is telling it that this is the original relaxed position in which you have to return. But how exactly is that determined? So you will observe this many a times, that our muscles have a particular relaxed posture in which they always return to when no force is applied, internally or externally. That gives us the information that there is some particular sensor involved which actually determines what the stretch or the contracted condition of the muscle is and where it should go once the force is removed. So these are done by the intrafusal muscles. These are essentially sensors. So what happens is that the intrafusal muscles are innervated via a particular group of nerves known as gamma nerves. So yeah, so these gamma. So now, first I should say that the interfusal muscles are innervated by a bunch of afferent nerve fibers. These nerve fibers are connected to the entire interfusal muscle, and when the muscle is contracted as a whole, the interfusal muscle also is contracted or relaxed. In that particular condition, the interfusal muscle starts sending signals back to the spinal cord and the motor cortex that the muscle is being stretched. This information is processed, and once the force is removed, the entire motor cortex starts the process of getting the muscle back to its relaxed state. There are two parts to this. First, it asks the alpha neurons, which innervate the entirety of the muscle, the extrafusal part of it, to start sending signals to ask the muscle to go back to its original state. So these alpha fibers essentially take the part of saying that the muscle that you were being contracted, now the force is gone, now you can relax back to its original state. And once this signal is transferred via the alpha neurons, the muscle comes back to its original position. but that does not pertain to our intrafusal fibers. These are still stretched. What happens is that there is a second set of neurons known as the gamma efferent fibers. These innervate the intrafusal fibers, and the information to relax is provided to the intrafusal fibers via these gamma fibers. So the gamma neurons tell the interneurons to relax, and the alpha neurons ask the extrafusal fibers to relax. So gamma intrafusal, alpha extrafusal, but both of them are essentially ask them to come back to its original position after a contraction has taken place. So the question was blank innervate extrafusal fibers and blank innervate intrafusal fibers. The correct option is B. Alpha motor neurons innervate extrafusal fibers and gamma motor neurons innervate the intrafusal fibers. Any questions from this? Like, I guess there are no questions on this, then we can move on. At the neuromuscular junction, the synapse is found between the presynaptic blank and postsynaptic blank. So what are the which part is presynaptic, which part is postsynaptic? The options are muscle and motor neuron, muscle and muscle, motor neuron and motor neuron, motor neuron and muscle. So which of these combinations are presynaptic and which are postsynaptic? Aruna says it is D. The motor neuron is presynaptic and the muscle is postsynaptic. Vishwajit also says that it is B. T, rather D, that is motor neuron is presynaptic and muscle is postsynaptic. Anyone else? Anyone else wants to try and answer this? Okay. So here regarding the neuromuscular junction, here as well we have a video. Let me go ahead and play it for you. Jagruti also says that it is D. Very nice. Just let me know if it is audible.
So when we're at a social gathering and we've determined that this is the most appropriate time to
Is this audible?
Yes, sir.
Okay.
perform the worm, how does our brain tell our muscles to contract? Well, in this video we'll talk about the place where neurons talk directly to muscles. That's the neuromuscular junction, the junction of where motor neurons talk to muscle cells. So that involves first the axon terminal. This is the end of an axon, which is the part of a neuron that casts a signal away, and it looks like this. It gets larger at the end right here, and then it kind of tapers back off like that. Muscle cells sit adjacent to these axon terminals at the neuromuscular junction and kind of look like a block, but not exactly. They have these infoldings that I'm drawing right here, these inpouchings. And why does nature cause inpouchings to occur? What's the purpose of these guys? What function do they serve? Well, if you said that it serves to increase surface area, you're absolutely right. Because with the increased surface area, we're going to have extra space where we can have sodium channels present that will help us transmit a message into the muscle cell. And so it's not just present on the outside, but there are a bunch of sodium channels that are deep inside as well. And in addition to sodium channels, you definitely have calcium channels that are present as well. They're situated very deep within your muscle cells too, to make sure that the most deepest parts of your muscle cells will get an influx of calcium when it's the right time. And to foreshadow a point we'll discuss later, I'm going to draw another muscle cell right here, just kind of chilling out on its own. So now, how does the axon send a message to the muscle cell? Well, if you recall, there's going to be a signal that's cast away from our motor neuron to this axon terminal, and that signal is in the form of an influx of sodium ions. So this is a depolarized membrane that propagates this signal to this axon terminal. But it's not just sodium that's influxing. You're also going to have some calcium that's running in as well. And the calcium here is actually going to play the major role, because in our axon terminal, we have a bunch of vesicles that are sitting around in here. These are just little pockets that are waiting for something to happen. And in each of these pockets, we have a message that's waiting to be released into the space between our axon terminal and the muscle cell. This message is called a neurotransmitter. Neurotransmitter, which is a very well-named scientific term, because all this is a molecule that the neuron uses to transmit a message. And so the neurotransmitter that we use in the neuromuscular junction is called acetyl, like from chemistry, acetylcholine. Acetylcholine. And oftentimes you'll see it abbreviated as ACH. So when there's an influx of calcium into the axon terminal, what will happen is that the calcium will actually bind to our vesicle. There are proteins that are on it that'll grab onto the calcium. And so when there's calcium attached to these proteins, the vesicle will be drawn to this axon terminal membrane and actually fuse with it to become one continuous membrane. As a result, we release the acetylcholine into this space we call the synaptic cleft. The synaptic cleft is just the space between our presynaptic membrane, the membrane of our axon terminal, as well as our postsynaptic membrane, which is just the membrane of our muscle cell. So this is our postsynaptic membrane. Alright, and so we have a bunch of acetylcholine that's released into this synaptic cleft and is ready to send a message on. But let's take a minute here. What just happened here with the membrane? I mean, the vesicle literally became one with the membrane of the axon terminal. What would you call this if you had to give it a name? Well, it looks like some compound within the cell exited the cell. So I'll say exo, and it exited a cell, so a site, exocytosis. And that's the process of molecules or substrates leaving a cell by vesicles fusing with membranes. And so that's what we do when we want things to leave cells. The exact opposite process, where we have things enter cells by fusion of vesicles, is called endocytosis. Endocytosis. And these are very important terms to keep in mind. Great. So now we've got acetylcholine all over our synaptic cleft. What is it going to do here? Well, these sodium channels have receptors that sit on them that are called nicotinic acetylcholine receptors. And as the name suggests, acetylcholine can come and very snugly sit here and send a message to this sodium channel that it's time to open and cause sodium to influx into our muscle cell. And that's going to happen across the membrane, causing a large amount of sodium to enter. And once we've depolarized the membrane enough, calcium will even start to enter. And so in this way, we'll have what's referred to as voltage-gated calcium release. Voltage-gated calcium release. Alright, and so we have all this calcium that's entering the cell. Now what? Well, this is just one cell contracting. How does this make me able to do the worm, or to kick a ball, or to high-five my best bro? Well, we can't just have calcium entering through the membrane. There's another reservoir within our muscle cells that releases calcium for our disposal. This reservoir, kind of a large name, and I'll write it out right here, is called the sarcoplasmic reticulum. RETICULUM. Sarcoplasmic reticulum. This guy holds a whole bunch of calcium. It's sitting in there waiting to be released, waiting to do something. So there are proteins that are attached to the membrane of the muscle cell, and they're just waiting for enough calcium to be present here so that one of these calcium cations could bind this protein complex and then effectively cause calcium to be released from the sarcoplasmic reticulum. This process is called calcium-induced calcium release. Calcium-induced calcium release. And so now we have a bunch of calcium in here, and this muscle cell will be contracting, but it's still just one muscle cell. What's the big deal? Well, this muscle cell is attached to its neighbor right here, actually. There are proteins that link muscle cells together. These proteins are called gap junctions. Gap junctions. And they allow for cations to flow from one muscle cell into another. So these muscle cells aren't really separate at all. Actually, they're connected because of this gap junction and actually continuous. And there's a term that we use to drive home the fact that we can have our calcium cations move to this other muscle cell and to start a calcium-induced calcium release process here. And it's that our muscle cells are in a syncytium. A syncytium. What does that mean? Well, site, just like we talked about here, just means a cell, and syn means that these muscle cells are in a synergy with each other. When one contracts, it causes its neighbor to contract as well. And that's how we scale up, because when muscle cells start contracting as neighbors, you get the entire neighborhood contracting, because you can then scale up and imagine that not just muscle cells, but muscle fascicles will also be contracting too, to produce a kick of a ball, or the worm. So this signal that started from our axon terminal here, that began as just depolarization, turns into an acetylcholine molecule that undergoes exocytosis to end up in the synaptic cleft, where it binds a nicotinic acetylcholine receptor to cause sodium to enter a muscle cell, and over time calcium to enter a muscle cell, which would then cause calcium-induced calcium release, which can then go to an adjacent muscle cell to cause a synergy of muscle contraction that spans from muscle cell to muscle cell, from fascicle to fascicle. And that's what happens at the neuromuscular junction.
So I hope that clears what exactly we mean by a neuromuscular junction. It's a very interesting bit of biology there, and I hope that we have understood it. We will just briefly summarize what was said in the entire video. So here we see a neuromuscular junction right here, in which the nerve innervates the muscle, and in this particular situation, acetylcholine, once the nerve is excited, acetylcholine is released into the synaptic cleft in the neuromuscular junction, and this particular synaptic cleft has various acetylcholine receptors. These receptors bind to the acetylcholine and allows exchange of cations and entry of cations into the muscle, which starts the entire process of calcium-induced calcium release. These calcium receptors are different from the ones that are present on the surface of the neuromuscular junction. These include the rhinodyne and the dipyridine, dihydropyridine receptors that we are talking about in the lecture series. And these calcium interact with the calcium binding to these particular receptors conveys a signal to the sarcoplasmic reticulum, which contains a huge store of calcium ions, which is then released into the body of the muscle fibers, where, as we have previously seen, that the calcium ions are the ones that drive the contraction. They allow the movement or removal of tropomyosin from the actin filaments, and once the actin filaments are free, they can cross bridge with the myosin and initiate the contraction process. So this is essentially what happens throughout the muscle cells. And then we have these T tubules, which we just saw, were indentations or deep channels into the entirety of the muscle. Since the muscle is such a thick tissue, it is very difficult for any action potential on the surface to reach deep into the tissue very fast, and we need almost instantaneous contraction throughout the entire muscle for any contraction to be actually feasible. So we have these T tubules, which are essentially tubes which connect the surface all the way to the depth of the muscle. Once there is a signal from neuromuscular junction, this signal can traverse the entirety of the depth of the muscle via these T tubules, and there the calcium sensing channels are present, which also further convey the calcium related information to the sarcoplasmic reticulum. So essentially at one go you can excite the entirety of the muscle using these T tubules. Is all of that fine? Anyone has any questions from the video or from this?
No, sir.
Okay. If there are no questions, then we go forward and answer the question. At the neuromuscular junction, the synapse is found between presynaptic blank and postsynaptic blank. The presynaptic structure is the motor neuron. The postsynaptic structure is the muscle. So the correct option is option D, as all of you were saying. Okay. So inactivation or blocking of acetylcholine receptors on muscles would cause which of the following? The options are overstimulation of muscle or tetanic response, no stimulation of muscle movement at all may lead to paralysis, normal muscle activity, there will be no change, both A and B. You can put whatever you think is the correct option into the chat. So inactivation of blocking of acetylcholine receptors on muscles would cause which of the following? So if the acetylcholine receptors, which were actually conveying the signal from the neuron to the muscle, were blocked, which of the following would occur? Vishwajit says it is B, no muscle stimulation and movement at all, and it may lead to paralysis. Anyone else? Anyone else wants to answer this? Okay, so we just saw the neuromuscular junction in detail. I will just go through it again. So here we have a neuron which is synapsing onto a muscle. At the end in the neuron itself, it is mostly acetylcholine or cholinergic neurons which contain the neurotransmitters acetylcholine. Once the neuron is activated or is showing action potential, and it reaches the axonal terminal, then various voltage gated calcium channels open up. These voltage gated calcium channels cause the entry of calcium ions into the neuron and allows the neurotransmitters filled vesicles to merge with the membrane at the end of the synapse. Once these neurotransmitters are released via merging with the synaptic terminal, then we have release of acetylcholine, which then will go and bind to various acetylcholine gated ligand receptors present on the muscle cells. These will allow the transfer of sodium ions into the particular muscle and initiate action potential in the muscle, and then muscle contraction will take place, as we have already seen. The essential part here is that the acetylcholine based neurotransmitters are very important to the entire process. If the receptors or the release of the ligands are hampered in any way, then we will not have any muscle movement, which will lead to paralysis. So inactivation or blocking of acetylcholine receptors on the muscle would cause no muscle stimulation and movement at all, and may lead to paralysis. Is that okay? Is that fine?
Yes, sir.
Okay. The next question is an excess increase in the release of acetylcholine receptors at the neuromuscular junction would cause which of the following? The options are overstimulation of muscle or tetanic response, no muscle stimulation and movement at all, may lead to paralysis, normal muscle activity, no change is caused, both A and B. Which it says it is A, overstimulation of muscle or tetanic response. Tetanic response means shaking of your hand, or essentially by overstimulation of your muscles. Anyone else wants to try and answer this? Okay, so the situation is very similar to a previous case where there is no stimulation. If there is excess stimulation of acetylcholine using agonist agonists are molecules which are very similar to our normal ligands but can activate our particular receptors nonetheless. And these agonists, if they go and bind to the acetylcholine receptors, they will cause overstimulation of the entire process, because they will keep the cation channels open to allow more and more cations to go in and cause a permanent or very high chances of a permanent action potential, so or depolarization. So the depolarization state will be maintained because no one is there to close these cationic channels. So overall it will cause a overstimulation of the muscle or the tetanic response. Is that okay?
Yes, sir.
Okay. So the next question is, so we move gears, change gears, and are now going to talk about one of the special senses that we have covered in this week's lecture series, which is the ear and how the auditory functions are done. So the first question from this is, what part of the ear contains the bones malleus, incus, and stapes? The options are outer ear, middle ear, inner ear, or there are no bones in the ear. Vishwajit says it is B. Middle ears is where the malleus, incus, and the stapes exist or are present. Anyone else wants to try and answer this? So if you look at a schematic diagram of the ear, you can essentially divide it into three major parts. First is the outer ear, which consists of the earlobe and the ear canal, which ends in the eardrum. At the eardrum starts the middle ear. Here we have the eardrum and three bones which are present in our ear and are the most smallest bones in our body, the malleus, incus and the stapes. They are arranged in a hammer and anvil position, so that whenever the eardrum vibrates, they essentially amplify the entire vibration and cause the next part of the inner ear to start vibrating even uh, more, essentially amplify the entire signal. Finally, the inner ear consists of the cochlea, which is the snail-like organ that you see right here and is responsible for the entirety of our hearing. So the stapes essentially is present at opening in the cochlea, and this particular organ is filled up with the liquid. The stapes essentially vibrates this particular liquid and causes the vibration to reach the inner ear, and this vibration is then detected via various hair cells present on the inner side of the cochlea, and these vibrations are then transferred as information to the auditory nerve, which is then processed by our auditory cortex in order to get the information out of the sound waves. So this is how exactly it works. We have a video again regarding the auditory or how the auditory complex works. Just let me know if
Look
you can
at
hear
the
this.
cochlea and the inner ear. Let me just draw a little cochlea. So
This is audible, right?
Yes, sir.
it's this round snail-like structure. But let's go ahead and unroll it. So let's just unroll it and lie it flat. So if we unroll it, it'll look something like this. It'll look something like that. So basically this is just a flattened cochlea. And so you've got this little bone that we were talking about earlier, which is called the stapes. So you've got this little bone right here. It's called the stapes, and this stapes is connected to the other two bones and then the eardrum. So it's basically moving back and forth at the same frequency as the sound wave that's making the eardrum move back and forth. And this stapes is connected to this little oval membrane called the elliptical window. And the elliptical window gets pushed in and out as the stapes moves back and forth. So as the elliptical window gets pushed in, there's fluid inside the cochlea. So we've got fluid inside, and that fluid basically gets pushed all the way around the cochlea and then comes back around. And as I mentioned before, the reason that the fluid flows in this direction is because there's actually a structure right in the middle. There's this little structure, and we call it the organ of Corti. So this organ of Corti kind of splits the cochlea in two, and the fluid can only flow in this direction. So when the fluid gets over here, there's this little round window, it's actually called the circular window, and the circular window gets pushed out a little bit as the fluid kind of compresses it. So now you've got fluid flow back and forth around this organ of Corti. So what we want to look at now is we just want to look at a cross section, just a little cross section of this organ of Corti, so that we can kind of understand what happens. How does it turn this fluid motion into sound? So if you actually kind of look at a cross section, what you would see is something like this. So something like this. So you've got an upper membrane and a bottom and a lower membrane, and you've also got these little hair cells. So you've got little hair cells with little shark fin looking things on the tops. So you've got these little hair cells, and basically as there's fluid flow around this organ of Corti, it goes like that, and then the fluid kind of comes around and goes this way. So as we have fluid flow, it actually pushes down on this membrane and pushes up on this membrane. So you can kind of imagine how this fluid flow works. So as this membrane gets pushed up and down, it actually causes these little hair cells to move back and forth. So they're moving back and forth, they're vibrating, and basically what we can do is we can blow up these hair cells, so we can blow them up and look at them in a little bit more detail. So it kind of looks something like this. So there's their shark fin part, and then there's the cell. So this shark fin part actually is called the hair bundle. Hair bundle. And these aren't actually hairs. What they are is they're just a bunch of little filaments, and let me just draw it in a little bit bigger. So if we were to look at just the hair bundle, it would look something like this. It would look something like this. And each one of these filaments is called kinocilium. Kinocilium. So basically, a whole bunch of these little filaments are attached to one another and they form the hair bundle. So each kinocilium is actually connected to one another by this little spring-like structure called a tip link. So it's a little spring-like structure, and each one is called a tip link. So it links the tips of the kinocilium. So if we were to actually look at a tip link, so let's go ahead and look at just this, the tip of this kinocilium. If we were to look at just the tip of this kinocilium, it would look something like that. And you've got the little tip link attached to it. So you've got this little spring-like structure attached to the tip of this kinocilium. And in fact, the tip link isn't attached to the kinocilium directly, but it's actually attached to the gate of a potassium channel. So there's the little gate right here. So this is the little gate of a potassium channel. And so as these hair cells, as the little kinocilii get pushed back and forth because the fluid is moving in the cochlea, as they get pushed back and forth, it actually stretches on this spring. So let's say that the kinocilium gets stretched. Sorry, we're going to use this color over here. Let's say that the kinocilium gets stretched. It actually kind of looks like this. So now it's getting stretched, and as it gets stretched, it actually opens up this gate. So as the potassium channel gate opens up, there's potassium outside that then flows into the cell. So you've basically got potassium out here flowing into the cell. And there are actually all these other little channels, calcium channels, that get activated when potassium is inside the cell. So now you also have calcium flowing into the cell. So the flowing of potassium and calcium into the cell basically causes the cell to fire an action potential. So it basically stimulates another cell, which is known as a spiral ganglion cell. And the spiral ganglion cell then activates another cell that is part of the auditory nerve, which then goes to the brain. So basically this goes to the brain. So this is what happens when a sound wave comes into the ear and then gets transmitted into a neural impulse by these little hair cells. In this video,
Yeah, so I hope that clears out any questions that were being asked about how the kinocilium and the stereocilium work together. If anything is there, then we can just discuss it after I summarize a bit here. Yeah, so these are what the hair cells look like. They have this tiny filamentous projections on top of them, which are known as the stereocilia and the kinocilium. The stereocilia is the first one that essentially faces the entire liquid part of the cochlea and will first get moved or about when there is any kind of movement of the liquid in the cochlea. So any sound comes in, this is the one that moves first. Once this moves, the entirety of the structure starts moving because they are connected via these transient links as well as tip links, which connect the tips of all of these. So, and how these function is that once the entire stereocilia and the kinocilium move in one direction, then this causes mechanosensitive calcium channels and cationic channels to open up and cause a action potential in this particular cell. This will be read via a neuron will be present right here and carry the information back to the auditory nerve. And here is what the cochlea looks like, the entire snail-like organ. Here you can see the hairy projections that are here. And the interesting fact here is that depending where the particular hair cell is, it can detect different frequency of vibrations. So the ones that at the furthest end or the nearest end to the states have the can detect the highest frequency, because in that frequency alone will they move about and cause a action potential in these cells. So any frequency this high will only be detected by the hair cells here. And as we move inside into the cochlea, we see that the entire frequency graph decreases. This is because these particular hair cells will only be detecting vibrations which are in lower frequencies. So the lowest frequencies are often detected by the hair cells which are at the deepest part of the cochlea, and higher frequency are often detected by the hair cells that are present at uh, the shallowest or the nearest part of the cochlea. Is that all okay? Any questions from this?
No, sir.
Okay. So the question, what part of the ear contains the bones malleus, incus and stapes? The answer is the middle ear. Okay, moving forward. Blanks are structures made of calcium carbonate in ear that sense gravity, tilt and acceleration. The options are the hair cells, the stale, the bone or the otoliths. Otoliths. You can put in whatever you think is the correct option into the chat. Which are the structures that help us to sense gravity, tilt and acceleration? Hair cells, stale, bone or otoliths? Okay, there are no answers incoming, so I guess that we actually do not know about this. So the inner ear, so this is the cochlea that we have already seen in detail. The inner ear consists of another structure, which is this huge structure right here, which are known as the semicircular canals. The cochlea are involved, as we have just seen, in the hearing process, but the inner ear consists of the semicircular canals which are involved in another process, which is maintaining the balance and determining the movement state of the body. So whenever you go round and round, like spin about, you feel dizzy. Why is that? That is because the inner ear can sense, has a sense of balance, which gets disrupted when you go round and round. So if you are sitting down or sleeping in a bed, you always are aware of your own spatial position as opposed to the ground. Yeah, so all of this is determined by the balance sensing organs present in the semicircular canals. So these are called the otoliths. So the entire canal is essentially in a semicircle like you see, and it contains the same liquid that is present in the cochlea. Once we are in a particular orientation, then the liquid fills only part of the semicircular canals which are detected, and the signal conveyed that you are either sitting down or sleeping or you're turning round and round, which causes all the liquid here to go haywire and causes the dizzy feeling, because your brain does not understand that it feels that you are completely out of balance because this liquid is moving around very fast. So the other function of the inner ear is to determine your balance. So blank are the structures made of calcium carbonate in ear that sense gravity, tilt and acceleration. The answers are the otoliths, which are present in the semicircular canals. Okay, so that was all I had for you today. Any questions regarding today's lectures or the entire session? Okay, so I believe that there are no questions. So with that, I would like to say thank you for joining today's session, and we will meet in the next session on next Tuesday, where we will be discussing further on neural system and special senses. Thank you everyone.